Hello everyone, my name is Brenda Sanmiguel-Mercado. I am from Mexico and I'm going to present evolutionary stability of receptor binding domain of SARS-CoV-2 spike protein. As introduction, since early 2021, more than 100,000 single mutations have been detected in SARS-CoV-2 genome worldwide. In silico studies have shown that a specific mutation in SARS-CoV-2 spike protein can change its structural stability and recognition affinity by the IC2 cell receptor. In particular, receptor binding domain of S protein is crucial to enhance the effectivity of SARS-CoV-2. Furthermore, any terminal domain of S protein contributes to changing AC2 RBD conformation, and its variability serves to evade the host in human response. Thus, our objective is to determine the evolutionary stability of the receptor binding domain of SARS-CoV-2 spike protein. As methods, we use a database from NCBI and preprocessing 39,457 complete sequences SARS-CoV-2 spike protein, and then with an alignment in error we see the amino acid changes in RBD motif, and after that with the algorithm in error we retrieve the fields of the first time each mutation was detected, and with Discovery Studio we performing each mutation and we can see the crystal structure of the receptor ligand. And after that with CLOSPRO we use a molecular docking where between each RBD mutation and the AC2 receptor. And finally, we make a sigmoidal model where the ligand receptor binding energy corresponding to each mutation date were plotted. As a result in discussion, we can see the amino acid variability in SARS-CoV and the SARS-CoV-2 RBD sequences. Here we have 206 SARS-CoV-2 and 174 SARS-CoV-2 sequences, and we can see a different variability in the RBD regions, and could be the region that is modified, mutated, to infect other species, namely that is crucial to in this evolutionary adaptation with the human host. Therefore, should be some combination of mutation that produces stable conformation between RBD and AC2. And here we can see the mutation frequency plot of variance shows where amino acid change occur. For positions and one region we see with the highest rate of point mutations: A 344, B 367, L 452, A 520, and the region A 475 to E 484. A region of maximum variability in RBD sequences and variability motifs represented in tertiary structure of RBD. The mutation observed in the position of the protein in the region 330 to 530 of the amino acid position, and on the other hand, the date on which each mutation was detected. We observe that there are conserved and variability regions, and the most important variability region is the one that is in green color, where the crest is located, which is the one that binds to the receptor, that according to the literature consulted, the crest is important for the union and detection on the target cell. The interaction energy and the date on which each mutation was reported are plotted. As the pandemic progresses, it is observed that the registered mutation increases the interaction energy, which is why the ligand becomes more related to the receptor. Our results suggest an adaptive stability trend that is associated with the higher affinity energy in RBD AC2 recognition along the timeline. This reflects an evolutionary adaptation that is independent of the geographical region in which mutation was aroused. As a conclusion, the genetic diversity of SARS-CoV-2 is likely to increase as it continues to circulate in the human population, so phenotype maps should become increasingly valuable for viral surveillance as mutations accumulate. The overall stability of RBD2 contributes to the binding affinity to AC2. In general, the mutational effects of RBD binding and expression are correlated to the high pressure of positive selection observed. This is leading to variants with higher affinity to AC2 for higher infectivity conjugated with antibody-mediated immune response evasion.